So let us start. So I welcome you all to the week 6 PMRF-NPTEL session on Introduction to Developmental Biology. My name is Anuvab Chatterjee and I am a PMRF fellow and PhD scholar under the supervision of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So these PMRF NPTEL sessions are designed in order to have an interactive problem solving session with the particular students who are opting for a NPTEL course and a PMRF scholar who will go through the various problem assignments of the previous year's run of the course and will discuss the various problems and how to approach them. These sessions are designed in order to for the students to have a better understanding of how to approach the problem and also understand the various concepts that were delved into in the previous week. So if everyone has gone through the lectures of this particular week, you will know that this week we have gone through various signaling pathways and also touched up a bit on ECM molecules, and this is what we will be discussing this today. So let us get right into it. So the first question is on the hedgehog pathway. So we have, if you will remember, we have gone through the hedgehog pathway, the RTK pathway, the wind signaling, not signaling, and apoptosis pathway, and as well as TGF beta signaling. So one of them is the hedgehog pathway that we will go through first. The first question is, what will be the consequence of absence of cholesterol to the hedgehog pathway? So if you have gone through the slides, you will know that cholesterol is very important to hedgehog pathway, and absence of cholesterol gives rise to various aberrant conditions, which may include difficulty in anterior posterior patterning of a particular organism. And this was also discussed with the help of an image of a cyclops goat, which due to inhibition or due to having a particular diet in which it was eating various inhibitors of hedgehog pathway such as gervine and cyclopamine, it had only one eye, but the next progeny had only one eye. So hedgehog pathway is very important for patterning, anterior posterior patterning, and thus, and as cholesterol is also very important to the hedgehog pathway, we will now look into what exactly absence of cholesterol does to the hedgehog pathway. So the options are, the receptor will not bind the ligand, so and the ligand cleavage to active form will not occur if cholesterol is absent. Ligand cleavage will occur constitutively, that is, the cleaved ligand will also the ligand will always be cleaved, or the ligand will diffuse too quickly. These are the four options in which, if there is no cholesterol present, the ligand will act in this manner. So this is more than one option correct, because this is obviously a box type of question. So I would like everyone to try and answer. Kavya says 1 and 2. That is, the receptor will not bind the ligand and the ligand cleavage to active form will not happen. Anyone else wants to try? Milanini says number 2. The ligand cleavage to active form will not happen. Okay, so let us go through what exactly the hedgehog pathway, what is the necessity of cholesterol and the hedgehog pathway. So hedgehog pathway is necessary for induction of particular cell types, boundaries between tissues, vertebrate, limb, and neural differentiation. How exactly the ligand is formed is, the hedgehog ligand is first cleaved and only the N-terminal portion is secreted. That is, the entire protein is not secreted. But before secretion, the N-terminal portion gets modified by cholesterol. That is, there is post-translational modification of the N-terminal portion of hedgehog, and that is what forms the active form of the ligand. Cholesterol modification is also essential for diffusion. So cholesterol helps in binding of increasing the localized concentration of the hedgehog ligand, and this will help for receptor and ligand binding, as increased localized concentration in a particular region will have more cells undergo the hedgehog pathway. And here we have a chick embryo, I think, in which the Yes, this is a chick embryo in which, via the technique in situ RNA hybridization, we have been able to see the various areas where regions where the sonic hedgehog molecule is expressed. So as we can see, the nervous system, gut, and limb buds is where majorly sonic hedgehog is expressed. So, as we have just gone through the slide, we know the answers to the question will be: in the absence of cholesterol to the hedgehog pathway, the receptor will not bind the ligand. As I said, the functional form of the ligand is the N-terminal portion that is changed by cholesterol or modified by addition of cholesterol. And so, in the absence of cholesterol, there will not be an active ligand that will bind to the receptor. Similarly, the second is also true. The ligand cleavage to active form will not occur. And finally, ligand will diffuse too quickly as there is no cholesterol to have it bound to the ECM and increase the localized concentration. There will be too quick diffusion of the ligand, and thus we won't see localized concentration of hedgehog. But I do have something to add to this particular slide. So the hedgehog molecule, the signaling was discovered in Drosophila, and there only one type of one molecule was present, and it was named hedgehog. But when they moved to the mammalian system, they wanted to discover the various types or the homologous molecules of hedgehog. So, what are the ligands that were there? So, there were exactly three hedgehog ligands that were found. So, in mammalian system, as we see in many a times, many of as we see in many times, whatever proteins are expressed in lower organisms, in higher organisms it is many other times the case that there are various homologous proteins present. So for an example, if you know the Hox genes, which are very important for patterning, in mammalian system there are 4 Hox genes. So similarly, in hedgehog, in Drosophila there was 1 hedgehog, in mammalian system there were 3. So these are the desert hedgehog, the sonic hedgehog, and so 3 hedgehogs. But I would like to draw your attention to the name of this particular hedgehog, which is very important for limb patterning, which is sonic hedgehog. Why do you think this was named like this? Anyone will try to answer that? And like, why is this particular hedgehog molecule named sonic? You can try. You can just put it in the tab. Like, why do you think the name is sonic hedgehog? You can even put it in the chat. Anything you think why it might be. The character Sonic, exactly. So I do not know if you know that the various there is a particular cartoon character known as Sonic the Hedgehog, and Sonic Hedgehog was named after that particular character itself. Yes, the other one. So the 3 homologous ones are Desert Hedgehog, Indian Hedgehog, and Sonic Hedgehog. And the story goes that when the homologous counterparts were discovered, it was discovered in America. And when they discovered the 3 of them, they tried, since in Drosophila it was named hedgehog, they tried to name it in as there were 3 species of hedgehog present in the world, which are desert hedgehog, Indian hedgehog, and European hedgehog. So they thought that they will name it desert, Indian, and European. But since it was found in America, they did not want to name it European hedgehog, the last molecule. So they named it Sonic the Hedgehog or Sonic Hedgehog. So that was a bit of tidbit that I wanted to add. So we move on and we go on to the next pathway, which is the JAK-STAT pathway. So the question is, how does STAT5 mediate signaling in the JAK-STAT pathway? The options are: it acts as a signal transducer, it acts as a transcriptional activator, it acts as a receptor, and it acts as a ligand. So this is also a box type of question. So how does STAT5 mediate signaling? So what is its function in the JAK-STAT pathway? Anyone will try to answer this? One and two. It acts as a signal transducer and it acts as a transcriptional activator. Kavya also says it is one and two. Signal transducer and transcriptional activator. Anyone else wants to try? Okay. So let us go right through the JAK-STAT pathway, and we have an example via the prolactin, where the prolactin receptor is shown, and how it activates the JAK-STAT pathway. So the answer essentially is in the name itself, because the name STAT stands for stat stands for signal transducer and activator of transcription. So the molecule STAT acts as a signal transducer. That is, it itself is it is the secondary messenger which actually goes into the nucleus and activates transcription. So we'll go through the pathway so we can understand it better. So the Janus kinase or JAK receptors are dimers which are not bound to each other in normal form. When a ligand such as prolactin comes and binds to it, the receptors dimerize and autophosphorylate each other. So we have phosphorylated JAK. Now what JAK does is it phosphorylates STAT, and STAT, which is a monomer normally in the cytosol, but once phosphorylated it goes to a functional dimeric state. So it undergoes dimerization, and we have STAT5 dimers. Now what happens is STAT5 itself goes into the nucleus and acts as a transcription activator and helps in transcription of various genes in response to the ligand binding to the receptor. In other pathways that we have seen, if you remember the RTK pathway, there are various steps in between these, and here STAT is acting as a secondary messenger, and it will then, if we go back to the RTK pathway, we know there are various steps such as RAS activating MEK, then there is ERK, and finally ERK goes into the nucleus and acts as a transcriptional activator. But here the secondary messenger STAT itself is activated and goes into the nucleus. And thus this is the speciality of this particular pathway, in which the secondary messenger is the transcription inducer, and that is where the name signal transducer, the one that is helping in conduction of the signal from the receptor to the cell, and activator of transcription, as it is itself going into the nucleus and activating transcription. So how does STAT5 mediate signaling in JAK-STAT pathway? It acts as a signal transducer and it acts as a transcriptional activator. So we move on. Next we look into juxtacrine signaling. So before we go into the question, can anyone tell me what exactly juxtacrine signaling is? There are 4 kinds of signaling. Can anyone name the 4 kinds of signaling? Like, what are the 4 kinds of signaling? One of them is juxtacrine signaling. What are the others? Autocrine, paracrine, and endocrine. Yes, Ujjwal is correct. Kavi is also right. There is another one that is called paracrine. And what is the difference between them? Like, not the difference. So you can try answering. Like, what is the specialty of juxtacrine signaling? Why is it named juxtacrine? Yes, Kavya is correct. The name of the various signalings were signaling pathways were differentiated on between the distance that between the signal transfer, or the distance the ligand has to travel. So, or in autocrine signaling, the cell that is producing the ligand, it is has receptors to the ligand itself. In juxtacrine signaling, the cells are very close by, and one cell that is touching the other is actually causing the signal to be transduced. Paracrine signaling is local signaling in which the ligand does not travel very far away, and endocrine signaling is where the ligand travels the entire length of the body probably. And the examples of endocrine signaling includes hormones. So in pituitary hormone has to travel all the way to the adrenal glands, and that is a very long range signal. So there is that. So the signaling that we just looked into, hedgehog and jackstat, what do you think, which category does it fall into? Paracrine, exactly. It is paracrine signaling. So RTK pathway, hedgehog pathway, and jackstat pathway, these are all TGF beta pathway. These are all paracrine signaling pathways that we had been discussing. The first, now we are just getting into juxtacrine signaling. So the question is, in juxtacrine signaling, I will, as I said, juxtacrine signaling, the cells are very close to each other, and there is direct contact between the cells, and this is what induces the signal from one cell to the other. So the question is, the juxtacrine signaling pathway functions in which of the following contexts? So juxtacrine signaling is important for which of the following? The options are vulval induction in C. elegans, lens induction through FGF8, vertebrate limb patterning, and fly and vertebrate nervous systems. So the question is, juxtacrine signaling is important for which of the following? Kavya says, Kavya, Renini and Ujjwal, all of them say 1 and 4, vulval induction in C. elegans and fly and vertebrate nervous system. Okay, let us look into the slides. So juxtacrine signaling or linked to a notch signaling. So notch is the particular signaling that we are talking about here. Notch signaling acts via the direct contact between two cells, and it is important for vulval development in C. elegans, as we have already seen. We had seen the example of how the vulva develops in C. elegans, and we saw how the P6 point, the P6 cell undergoes produces a lateral inhibition towards the cells on either side of it, so that the P6 cell can undergo the primary differentiation and have primary fate and form the vulva itself, while P5 and P4 cells on either side of it do not, and they undergo the secondary fate. So that is what seen in the vulva development in C. elegans, and we also see it in fly and vertebrate nervous system development as well. And it determines which of them become the optic neurons and which of them become glial cells in the nervous system. So we will go through quickly how the notch delta signaling works. The receptor for notch is notch itself, while the activator is the delta protein that is shown in the second picture. In normally, notch is present as a transmembrane protein where there is a small disconnect or disjunction between the transmembrane domain and the cytosolic domain. When delta is present, that is the signaling ligand itself, it binds to the extracellular part of notch, signals to the cytosolic part of it, so that a particular protease is now able to cleave the cytosolic domain of notch, and it now goes directly into the nucleus and activates transcription. So this is how notch delta signaling works. So the juxtacrine signaling pathway works in the following ways: vulval induction in C. elegans, fly and vertebrate nervous system, and lens induction through FGF8. I don't think it will be lens induction through FGF8. I will look into it. But the 1 and 4 is correct. I will look if the second one is correct or not. But 1 and 4 is the correct answer. So we go on to the next question. Which of the following signaling pathways involve positive feedback effect on production of the receptor? So which of the following pathways acts via positive feedback signaling loop in order to produce various effects? The options are notch pathway, jack-stat pathway, wind pathway, and the apoptotic pathway. This is a circle type of question, so only one of them are correct. A positive feedback loop is observed in, Kavya says number 1, the notch pathway. Mrinalini says number 2, the jack-stat pathway. Anyone else wants to try this? Okay. So first I think we have to go through what exactly we mean by positive feedback. The answer to the particular question, positive feedback is seen in the notch pathway, the notch delta pathway. But what exactly happens is, when, as I said, the for notch pathway, since it is a juxtacrine signaling, the two cells have to be very close to each other. And this is a particular example that we are going through, in which there are two cells which are in an equivalence group. What equivalence group means is that a population of cells which could have undergone various steps of differentiation and formed and could have had a similar fate but did not. So the two cells we are talking about are the anchor cell precursor and the ventral uterine precursor. These two cells could have formed each other as well. So the right cell could have formed the anchor cell and the left one could have formed the ventral uterine precursor cell as well. But due to notch signaling, one only differentiates to the anchor cell and one only forms the ventral uterine precursor. So how does it work? So these two cells are named the Z1 PPP, the Z1, the posterior daughter of Z1, and Z4 AAA, the anterior daughter of Z4. What happens is that both of them have notch and delta ligands and receptors present on the surfaces. One of the downstream targets of notch signaling is production of excess amount of notch itself. So what happens is that a particular cell may be due to abrupt stochastic increase, so very minute changes in mRNA level or translational level produces a higher amount of notch. Since one cell produces higher amount of notch, and therefore when it produces the notch signaling in the left cell, the right receives higher amount of notch signaling, and this signals it to form greater amount of delta. This signal now gets back to the previous cell and it gets a positive feedback to form more notch, and this continues till the right cell gets the positive feedback. Okay. Hello. Hello. Am I audible now? Hello. Okay. So we will go back to that part again. So this is what I was trying to tell you is, in this particular example, there is a left cell which is producing a lot of signal, a right cell which is producing a lot of receptor. When notch signaling in the left cell increases, so when the notch signaling in the left cell increases, it produces a lot of notch on its surface. So it is signaling to the right cell to produce a lot of delta. This is a feedback loop. So the left cell tells the right one to form more delta, The right cell for it asks the left one to form more notch. This goes back, continues for some time till one of the cells is only expressing notch, one of the cells is only expressing delta, and not the other way around. And this is the signal for the particular cell to differentiate into a specific fate. And so the cell which is only expressing notch will form the anchor cell. The cell which only expresses delta will form the ventral uterine precursor. This is also seen in nervous system development in various organisms, in which there is a population of cells which can form nerves or can also form glial cells. But due to these stochastic changes, only one particular cell ends up expressing notch, and every one of the neighboring cells only expresses delta. And the one which expresses notch is one which one finally forms the neuron, and every one of the other cells form the glial cells. So this is exactly how notch delta signaling practically occurs, and this is the function of notch delta signaling. And thus, which of the following positive feedback effect on the production of the receptors is notch signaling or the notch pathway. Anyone wants to ask anything from this particular slide? Any confusion? Anyone wants to get clear? I guess not. Okay. So we move on to the next question. Vulval development in C. elegans is regulated by which of the following pathways? The pathways are notch pathway, hedgehog pathway, TGF beta pathway, and RTK pathway. This is something that we have gone through in the previous slides, in the previous lectures, or how exactly the vulva develops. And we had in those particular lectures, we had gone through various proteins or genes such as Lin12, Lin3, Let60, which we had said okay, these genes are responsible for a particular function, but we never actually went into and understood how exactly the gene undergoes that function. In this particular question, we'll now understand which gene is responsible for which signaling pathway and how exactly it forms the various parts of the vulva. So this is a box type of question. So there is more than one option correct. So in vulval development, which of the following signaling pathways are active? Notch pathway, hedgehog pathway, TGF beta pathway, and RTK pathway. Anyone wants to try and answer this? Mrinalini says 1 and 4, notch and RTK pathway. Ujjwal also says 1 and 4, notch and RTK, and Kavya also says 1 and 4, notch pathway and RTK pathway. Anyone else wants to try? Arup also says 1 and 4. Okay. So let us look into how exactly the notch and notch pathway and the RTK pathway act in C. elegans vulva development. So first I will draw your attention to the left picture. So this is a picture that we have seen again and again, in which we have an anchor cell followed by a group of equivalent cells which are the PNP cells, and the P6.P cell, which is gets the most signaling from P6.P as shown here, will undergo the primary fate and form the vulva, while the neighboring cells P7.P and P5.P will form the secondary fate. So and every other cell which is not receiving any signaling from the anchor cell will form the hypodermis or undergo the tertiary fate. So we had already looked to the genes that were responsible for this signaling. These include Lin3 for the signaling between the anchor cell and the middle PNPs, and since the P6P, P5P, and P7P, all of them receive the anchor cell signaling, which is RTK signaling. So Lin3 is the RTK, or RTK undergoes is part of the RTK pathway, and the ligand itself is a FGF ligand. And since all of these three get the particular signaling, all of them could have undergone primary fate. But what happens is that P6P via juxtacrine signaling, that is not signaling, inhibits the P5P and P7P from undergoing the primary fate, and then they go into the secondary fate. So there is two levels of signaling that is occurring here. Between the anchor cell and the middle PNPs, we have RTK signaling, and within the P6P, P7P, and P5P, in order to determine which cell forms the primary fate and which forms the secondary fate, we have the notch signaling. So Lin3 is part of the RTK pathway, and Lin12 that we have already discussed is also a part is part of the notch delta signaling. So this is how exactly the vulva forms, and thus the answer to the particular question, the vulval development in C. elegans is regulated by the notch pathway and the RTK pathway. It's okay. Anyone wants to ask anything? Okay. So the next question is, which of the following is false with respect to vulval development in C. elegans? So which of the following is false? So we have just looked into vulval development. So now we will actually see which of the following, try to understand which of the following is the false answer. Okay, Biryani goes and answers. Let me read out the options first. The first option is vulval precursor cells form an equivalence group. Without the anchor cell, only P8P, P4P, and P3P attain tertiary fate. Lateral inhibition functions through notch pathway, and lateral inhibition functions through RTK pathway. Biryani says 2 and 4, that is, without anchor cell, P8P, P4P, and P3P attain tertiary fate, and lateral inhibition functions through RTK pathway are false. Arup says number 2 is false. Ujjwal and Kavya say number 2 and number 4 is false. Okay. So, as we have just looked into the slide, we know that the vulval precursor cells form an equivalence group that we have just discussed. All of the cells could have formed any of the fates. Without the anchor cell, only these three cells will not have had tertiary fate. All of the cells would have undergone tertiary fate. So that is correct. That is false. The lateral inhibition functions through notch pathway. This is correct. And lateral inhibition functions through RTK pathway. This is false, that we have just seen. So number 2 and number 4 are false answers. And thus, this is the how we will mark it. Anyone wants to ask anything from this particular question? Like this particular explanation was enough, I guess, on how exactly which cell forms undergoes which fate and what the signaling is. Yes, okay, nothing. Yes, okay. Okay, so we will now move into, from the signaling part of it, we'll go into the ECM, the discussion on ECM molecules, on how exactly ECM helps in various signalings. And the first question is, the RGD domain is present in which of the following proteins? Integrin, vinculin, tallin, and fibronectin. This particular discussion is on how exactly the ECM and ECM molecules and ECM proteins help in the signaling and the actin cytoskeleton rearrangement in various cells. So Ujjwal answers, the answer is 4, fibronectin. It is a one option correct question, so I would like anyone else to also try and answer this. The RGD domain is present in which of the following proteins? Anyone else wants to try? Okay. Kavya also says number 4, fibronectin. Okay. So let us look into the proteins themselves. So fibronectin, Sangeeta says 2, which was vinculin. Okay. So this particular diagram shows us various proteins that are involved in various proteins that help in the signaling between the ECM and the cell's actin cytoskeleton. We have fibronectin, which is a ECM protein that is present majorly in the ECM, And we have various proteins that help in the signal transduction from the ECM fibronectin towards the actin cytoskeleton of the particular cell. These include integrin proteins, which have an alpha subunit domain where there are calcium binding sites, and a beta subunit domain, which help the binding of the fibronectin to the another protein named tallin, and via and tallin and vinculin together help in the signal transduction from the fibronectin towards the actin cytoskeleton. And as you can see right here, fibronectin has various RGD domains, which is recognized by the beta subunit of integrin, and that is where the beta subunits bind and helps in signal transduction. Thus, so the integrins are a complex. The integrins of the complex are membrane-spanning receptor proteins that bind fibronectin on the outside of the cell while binding cytoskeletal proteins on the inside of the cell. Thus, the RGD domain is present in the protein fibronectin. So the answer is fibronectin. Vinculin, on the other hand, helps in signal transduction via tallin and does not have any RGD domains. Okay. So we move on and ask the question, which the integral membrane protein integrin acts as a receptor for which of the following molecules? So which of the following molecules will act as a receptor for the protein integrin? The options are collagen, actinin, fibronectin, and cadherin. So integral membrane protein. So what integral membrane protein means is that there are particular proteins that are present in the cell membrane. The proteins present in the cell membrane are divided on two parts. One of them is known as a peripheral protein, another is known as an integral protein. Peripheral proteins are present, which are present on just one side. So they have a transmembrane moiety that binds to the cell membrane, and another domain which either can be extracellular, depending if it is an extracellular peripheral protein, so it will have an extracellular part, or if it is an intracellular peripheral protein, then it will have a transmembrane domain and will have another domain which is inside the cell intracellularly. Those are peripheral proteins. Integral proteins are proteins which go right across the cell membrane and essentially have extracellular domain, a transmembrane domain, and an intracellular domain. So this is what is known as integral membrane proteins. So can anyone give me an example of an integral membrane protein? So proteins which essentially connect the extracellular part and the intracellular part of the cell. Anyone can try guessing what these proteins look like what can be an example of these integral membrane proteins? Hello? No one wants to try and get a guess? Take a guess. Okay, so have you so integral membrane protein, one of the major examples include protein channels or channel proteins, which help in conduction of various substances from the extracellular part of the cell to the intracellular part via their lumen. These can majorly include ions such as sodium, potassium, and calcium to pass through the membrane, as the membrane itself is non-polar in nature, so ionic molecules cannot pass through, and they need protein channels through which they can pass through. So these are what integral membrane proteins are. But now returning back to our question, the integral membrane protein integrin acts as a receptor for which of the following molecules? And the four options are collagen, actinin, fibronectin, and cadherin. So this is a one option correct round type of question. So I would encourage everyone to try and answer this. Kavya says it is 3 fibronectin. Ujjwal says it is 2 actin. Anyone else wants to try? Sangeeta also says it is number 2 actin. Okay. So the answer again is fibronectin. We will, if we go back to this particular diagram, we see this is fibronectin and integrin is the receptor which binds to fibronectin. So its beta domain binds directly to fibronectin, and its alpha domain has calcium binding sites, both of which signal back to the cell. So integrin here acts as a receptor, and the ligand or the protein to which it binds is fibronectin, not actin. So actinin is another cytoplasmic protein which acts via directly binds to the actin microfilaments. So the integral membrane protein integrin acts as a receptor for fibronectin. Is that okay? Anyone wants to add anything to this? Okay. So we move on and we go into the next question. Extracellular calcium regulates cell basement membrane and cell-cell interactions by binding to which of the following particular subunits of integrin or collagen. So extracellular calcium binds to what? Which of the following? Which of the following? The options are alpha-actinin, alpha subunit of integrin, beta subunit of integrin, and collagen. So Kavya says it is number 2, alpha subunit of integrin. Anyone else wants to try? Sangeeta says it is beta subunit of integrin. Extracellular calcium binds to beta subunit of integrin is what Sangeeta is saying. Anyone else wants to try? Okay. So we went too far away. Okay, so yeah, so here we can see that calcium binds to the alpha subunit of integrin before binding to fibronectin. Beta subunit only binds to directly to fibronectin via the RGD domains, and integrin is modulated or regulated by calcium binding sites on the alpha subunit. So the answer to the particular question, extracellular calcium regulates cell membrane, basement membrane, and cell-cell interactions by binding to the alpha subunit of integrin. Here we can again see this. Okay. Anyone wants to add something? So essentially, you can understand integrin as it has three. It has two. So it is a heterodimer in which it has a alpha subunit which has regulatory sites and a beta subunit which binds to the particular ligand. So the beta subunit binds to fibronectin, which is the ligand, which acts as a ligand, and alpha subunit is where the regulatory calcium binding sites are. And using these, the action of integrin can be modulated. So we move on, and now we look into following question, which is the principal role of proteoglycans of the ECM is. The options are delivery of paracrine factors to the respective receptors, organization of ECM into an ordered structure, mediation of epithelium basal lamina attachment, and mediation of mesenchyme fibronectin attachment. So first let me give you a bit of context. Proteoglycans are. Proteoglycans are large sugar molecules which have a very small proteinaceous component. The other very similar sounding name is glycoproteins, which are large proteins which have a small sugar modification such as glycosylation or something similar. Thus, proteoglycans are sugar molecules with protein part, and glycoproteins are protein molecules with sugar part. So the question is, what is the principal role of proteoglycans? The delivery of paracrine factors to their respective receptors, organization of ECM into an ordered structure, mediation of epithelium basal lamina attachment, and mediation of mesenchyme fibronectin attachment. So this is a round type of question, so only one of them are correct. So anyone wants to try and answer this? Kavyas is 1. Delivery of paracrine factors to the respective receptors. Anyone else wants to try and answer this? Okay. So let us look. So this here we can see an example of the ECM. Here this is an amphibian embryo, in which the fibronectin is marked out in green, as you can just see here, and this is part of the ECM. So ECM is highlighted, in which fibronectin is shown in green, and here is what the ECM looks like. So what we have is a cell of a layer of epithelium cells which secrete a particular basal lamina, which essentially acts as a base on which every one of the epithelial cells are attached. And under this exists a layer of mesenchymal cells, which secrete their own ECM, which is in the form of collagen. So basal lamina looks very uniform, is like a floor. Essentially, which is the ECM secreted by the epithelium, whereas collagen, which is the ECM secreted by the mesenchyme, is in this form, not very uniform. And this is also the point, because mesenchymal cells do not undergo attachment and do not need a very solid ECM. So what are the roles of the ECM in various contexts? The major role of ECM is it is a source of critical developmental signals. So we have seen how various signaling molecules are produced in the various cells, and those are secreted. But these, you have to imagine these as they do not travel just like that. They travel through the ECM, and thus the ECM is essentially the source of the critical developmental signals, especially for the paracrine signals. ECM proteins are important for delivery of paracrine ligands as well. So you can imagine that if a particular cell is undergoing the hedgehog pathway, it is producing sonic hedgehog, and then allowing it to be exported to the extracellular space. The first thing it encounters is the ECM, and depending on the nature of the ECM, particular regions of the ECM can actually attract and bind to and increase the local concentration of the particular paracrine ligand, such as sonic hedgehog, and thus the cells around that particular region will only be allowed to undergo the hedgehog pathway, because every one of in all the other places, even if there is hedgehog present, it is diffusing too fast away or not staying around the cell long enough for it to activate the signaling. So ECM proteins are extremely important for delivery of paracrine ligands, in which they allow for increase or for diffusion and for selective diffusion of various paracrine signaling ligands, and help specific cell population to undergo the particular signaling. ECM is also critical for cell attachment, migration, differentiation, and morphogenesis. This particular point was highlighted in the lecture slides using the example of mammary gland cells, and how exactly they only for produce they only undergo differentiation to form the glands when the given particular ECM to bind to. If you will remember, the cells are present normally present in the as a sheet, and when given the specific ECM required, they will fold and form lumen-like structure. So they are also critically important for cell attachment, migration, differentiation, and morphogenesis. Thus, the principal role of proteoglycans in the ECM is delivery of paracrine factors to their respective receptors. Next, we look into which of the following is or are examples of EMT or epithelium to mesenchymal transition. The options are development of mammary gland, formation of vertebrae from somites, formation of neural crest cells, formation of mesoderm in developing chick embryos. Which of the following is an example of EMT? So EMT is something that I think we had just touched upon in the last session, which is epithelial to mesenchymal transition, in which epithelial cells which are present on top of the basal lamina very in a very uniform layer attached to the cell neighboring cells decides that it wants to form a mesenchymal cell, and at that moment it dissociates its particular adherence junction which is binding to the which is what is binding it to the neighboring cells, burrows through the ECM. So it dissolves the ECM via various proteins which include MMPs or metalloproteases, and just burrows right through the ECM and goes underneath the basal lamina, and there it can exist as a mesenchymal cell. This is what epithelium to mesenchymal transition means. This is extensively studied in metastasis, as cancer cells in the later stages do undergo this particular process in order to travel to various other parts of the body and produce secondary cancers there. So the question is, which of the following is or are examples of EMT? The options are development of mammary gland, formation of vertebrae from somites, formation of neural crest cells, and formation of mesoderm in developing chick embryos. So this is a square type of question, so more than one is correct. Kavya says 2, 3, and 4. Formation of vertebrae from the somites, formation of neural crest cells, and formation of mesoderm in developing chick embryos. Shian says 2 and 3. Formation of vertebrae from somites, formation of neural crest cells. Anyone else wants to try? Okay. So let us look into how exactly the entire thing occurs. So we have a diagram showing us how exactly EMT occurs. So this is the basement lamina that we are talking about. This is what the epithelial cells secrete, and on top of this, these are the epithelial cells, which have a very defined morphology, have adherence junctions present between them so that they can bind to each other. So we can see various proteins that we have already looked into: cadherins, actins, catenins. So every one of them are joined together in a layer, and these also have an apical polarity complex. That is, there is a basal site, there is an apical site. But signaling from various paracrine factors can cause one of these cells to think that it wants to go into wants to form a mesenchymal cell. So it undergoes EMT. So the first stages include the signaling. The second is the cell adhesion to the neighboring cells are broken. So the junctions are actually broken, and the basement membrane is starting to be dissolved using various proteases, so that it can burrow right through the basal membrane, and finally the cell is released from the basement membrane and goes into the ECM complex itself, away from the epithelial cell layer. So these are still epithelial cells, and this particular cell has undergone EMT. This is, as I said, studied in metastasis and the pathogenic states such as cancer, but this is also very important in various other contexts as well. Remember, we had looked into neural crest cells. These are pigment producing cells as well as cells which form various parts of our craniofacial regions. So these cells are actually part initially part of the neural tube to the dorsal part of the neural tube. These are initially epithelial cells which undergo EMT and start forming mesenchymal cells. And now these neural crest cells, having taken the mesenchymal character, can travel, and they travel to various parts of the craniofacial regions and essentially form pigment producing cells there and various other structures such as nose or various parts of the skull. So they help a lot in forming these structures, and this is also relevant in formation of the mesoderm layer. So if you also remember in the very first lecture, I think we had looked into how the cells, in order to form the mesoderm and the mesoderm, first there are two layers of cells which are the epiblast and the hypoblast. The epiblast cells then travel via the neural crest and go into the embryo, and then we have the formation of the trilaminar disc which forms the three germ layer: the ectoderm, mesoderm and the endoderm. So I would request everyone to go through, and there were, like, I think we looked into videos of it, so it will be very easy to look into it if you want to. So this particular formation of the three germ layers also occurs through EMT, in which the epiblast has epithelial like cells which undergo EMT and then, forming mesenchymal characteristics, actually travel via the neural crest into the embryo to form the three layers. So here we have everything explained. Normal epithelial cells are attached to one or other through adherence junctions containing cadherin, catenins, and actin rings. They are attached to the basement membrane through integrins, as we had just seen, via the fibronectin connection. Paracrine factors repress the expression of genes that encode these cellular components, causing the cell to lose polarity, that is the apical basal polarity that it had, lose attachment to the basement membrane, and lose cohesion to the neighboring epithelial cells. Cytoskeletal remodeling occurs, as well as secretion of proteases, extracellular matrix molecules that enable migration of the newly formed mesenchymal cell. And this is the example that we have seen EMTs in vertebrate embryos during normal formation of neural crest from the dorsal region of the neural tube, and during formation of mesoderm by mesenchymal cells delaminating from the epiblast. Here there are other examples: formation of neural crest, formation of mesoderm in chick embryos, formation of vertebrate precursor cells from somites, wherein the cells detach from the somite and migrate around the developing spinal cord to form the vertebrae. So if you remember, somites are the way are embryonic structures that are formed on either side of the neural tube, and they will give rise to the vertebrates and the muscles in our back that support the spinal cord. And in there also there is EMT, which allows the somites to release cells which actually migrate around the spinal cord to form the vertebrate column. Okay, I guess that is fine. Like, everyone now understands what EMT is. Yes. Yes. No. Maybe. Anything? I am audible, right? Hello. Hello. Okay. So anyone wants to try, everything is okay, right? You understand what EMT is? Yes, no, maybe, anything I will do. Okay. So I guess that is a yes. So we go into and look into which of the following is our examples of EMT. These are formation of vertebrae from somites, formation of neural crest cells, and formation of mesoderm in developing chick embryos. Okay. So we move on to the next question, which is: identify the permissive ways in which cells maintain their state after differentiation. So this is something that we had, which was a part of the last lecture that we had seen, last part of the last lecture, in which, say, after a particular cell has gotten particular signals, and now the cell has to cells undergo replication and forms various cell population. But say a cell was about to form neurons, the daughter cell or that cell itself, after the signaling is over, has to still maintain and activate particular genes. It can't be like only when the signaling is present, then only these particular genes are present. Many important developmental genes, say, which are very important for neuron formation, needs to be activated throughout the life of the neuron. And but the signaling may not be present throughout the life of the neuron. So there must be particular ways in which, even after the signaling is absent, those particular genes or the gene products are being expressed. So what are these various permissive ways in which the cells can maintain their state of differentiation even after the signaling is gone from that particular region? So the options are: they maintain euchromatin state so that genes are always accessible. Euchromatin means that the chromatin itself is loosened up and opened up so that various transcription factors can go and bind to it and express the particular gene. And another way can be the gene product acts as an activator for its own expression. So what this means is that the after the gene is formed, the gene itself acts as a transcriptional regulator for itself. So more of the gene produces more of itself. The third option is neither A or B. And the fourth option is both A and B are ways in which cells maintain their state of differentiation. So this is a round type of question. That is only one of the options are correct. Kavya says the option is 4, both A and B. Anyone else wants to try? Okay. So Arul says it is number 4, both A and B. Shian says it is number 1, maintain euchromatin state so that genes are always accessible. Okay. So we'll look into it quickly, what exactly are the methods via which the state of differentiation is maintained, and there are 4 methods in total. So let us go into it one after the other. So once the first method is, once the initial activation stimulus is received, that the first signaling has occurred, and a particular gene which is from this promoter site, +1 site is where the gene is being expressed, the signaling acts on enhancer number 1 of the gene and allows the transcription of the particular gene. Once the gene is activated, gene product is produced, the gene product itself acts as a transcription factor and produces more of itself. This is the first option. So the gene product itself allows for production of more of the gene product. The second option is, after the initial stimulus from the extracellular ligand, there can be a particular complexes such as the trithorax complex, which binds to the promoter, and once it binds, it does not allow the DNA to refold again to form the heterochromatin structure. That is, the nucleosomes bind, and the nucleosomes come into place, then the histones bind, and we have a coil structure. But in this particular scenario, proteins such as the trithorax protein bind to the promoter so that it is always accessible to the RNA transcription machinery, so that the gene is always expressed. The third option is, the particular after the initial stimulus, the particular enhancer produces a gene product, and gene product itself is the ligand. So for example, we can take the hedgehog pathway. In the hedgehog pathway, the one of the downstream targets of hedgehog itself is patched, and in also other scenarios such as we see in notch delta signaling, as we have seen, one more production of notch, more notch signaling in particular cells allows for more production of notch on its surfaces. So the particular ligand itself is produced as the gene product after the signaling pathway, and it then travels out of the cell and acts on the particular its own particular receptors to form to continue the feedback cycle. So this is essentially autocrine signaling that is occurring here. And finally, we have this particular third example, but when two neighboring cells perform the same, in which after the signal is received by cell A, it produces the ligand, and the ligand now travels to this cell B, and cell B again produces a ligand for cell A. So essentially a feedback loop such as this, but in this case two cells are involved. This is also seen in various developmental paradigms, such as formation of hair follicles on the surface of the Drosophila embryo, is seen in a pathway a bit more complicated, but it follows the same scenario such as this. So, four ways in which differentiation can occur. This is the initial stimulus activates enhancer 1, which is the first scenario they are talking about, and the gene product activates binds to the enhancer 2 and then allows the production of the gene. Similarly, the trithorax complex is explained here, autocrine signaling as we just talked about, and finally the paracrine loop. So these are the four ways, and thus identify the permissive ways in which cells maintain their differentiation. These are both A and B, in which they maintain chromatin state so that genes are always accessible, and the gene product acts as an activator for its own expression. So that is all we had for today. We have gone through the signaling pathways, the functions of the ECM, and also touched upon how the particular signaling can be maintained in a particular signaling state can be maintained in cells even after the signaling is removed. So the references to all the slides are Introduction to Developmental Biology, the 9th edition by Scott Gilbert. This is the YouTube channel link on which we are uploading the various videos that are being recorded. So does anyone have any questions regarding today's presentation? If not, then if there are no questions, then we can end the session right here. Thank you everyone for joining today, and see you next week. Thank you everyone.